Having a good start to the week. A little warm in here today, I apologize. I don't know what's going on with our climate control, but hopefully someone will come and fix that for the rest of the week. So today, we're going to start where we finished with glycolysis and go all the rest of the way to cellular respiration. We're going to talk a little bit first about fermentation as the alternative to cellular respiration. First, some announcements. The exam is a week from today. I posted the study guide this morning. If you haven't seen that, I will get the Mastering Biology questions up this afternoon if you have access to that. For the exam, we're going to go as far as we can get through all these pathways by Friday. Then we also covered a little bit of genetics and membrane transport. All right, so this is a big section. Don't wait till this weekend to start looking it over. Start learning your pathway. We're all the way through glycolysis. You need to know all of the steps for that, all of the structures. Everything else you won't need to know in quite such detail. But remember that you need to keep track of where we're getting our ATP, where and what we're producing along the way. So for review for this next exam, your recitation section this week will be a review section. Instead of one giant review section, your recitation is going to serve for that. So everyone should have access and be free for at least one review session, yeah? And that way you'll have been through most of the material by the time you have recitation, hopefully, so you can have questions ready for your TIs. As far as office hours this week, unfortunately, I will not be able to have office hours today. I have some training that I have to go take. So I will have office hours on Wednesday. If you would like to come and talk to me some other time, just like always, just send me an email and we'll schedule a time to make that happen. If you want to come and ask me questions, if you're not clear about something, and you can't make Wednesday, let me know. Okay? I'll send out an announcement as well when I get back to my office, but no office hours today. Any questions related to the exam? Then let's keep going with cellular respiration. All right, you've seen this a number of times now. The 4 steps we're going to cover related to glycolysis and cellular respiration. You need to keep track of where things happen. So far we've been talking about glycolysis, which happens out in the cytosol. Today we're going to take that pyruvate and move into the mitochondria and finish the rest of the 3 linked pathways. This is what we have been going over the last few lectures. Taking glucose into the cell, what happens to it once it gets into the cell. Remember that there are 3 phases to glycolysis. That energy investment phase, the phase where we strip off that first NADH, and then going from 1,3-bisphosphoglycerate all the way to pyruvate. So remember at this point, we have 2 molecules of pyruvate. So everything we talk about happens twice. So on the exam, when I ask you for how many ATP have been produced up to this point, read the question very carefully. Am I asking for a single molecule, or do I want the product of everything that's been produced so far? Okay? So I need to make that clear for you to be able to tell what the right answer is. But you also need to read the question carefully about whether I'm asking for a single molecule or everything that's being produced from one molecule of glucose. If I start with glucose, Glucose gets broken down to 2-pyruvate, and you need to count everything twice. If I say start with 1 molecule of pyruvate, or what's produced from a single cycle of the citric acid cycle, that's a different question. Okay? So be sure you read the questions carefully. And that'll tell you how to add things up. All right. So, we'll start off. I'll give you the chance at this question again. The clicker had frozen by the time we got into this on Friday. All right, so this is leading up to what we're going to talk about today, and the importance of NAD plus and NADH to this whole process. So we talked all the way through glycolysis. We get NADH out at that second step, that second phase, where we go from G3P to 1,3-phosphoglycerate. What happens if we run out of NAD+. Alright. Everyone get their answers in? Are there really that few of us today?
Maybe it's the rain.
It's the rain. It's Monday morning, so the clicker is just not working in general. Oh, no one wants to come because it's raining. That could be as well. All right, I'll give you one more second to get your answers in. Let's take a look. All right, so we covered this at the very end of lecture on Friday. Without a fresh supply of NAD+, glycolysis stops. It can't go any farther. So NAD+ is limiting to this whole process. Without it, we can't go from G3P to 1,3-bisphosphoglycerate. So you just get the accumulation of G3P. Glycolysis stops. Can't go any farther. So regenerating our NAD+ is important for keeping the cell in a steady supply of energy. It's required for that second phase of glycolysis. So let's look at how cells regenerate NAD+. So there's two general pathways, two types of pathways that cells take. It can either undergo fermentation, which is an anaerobic process. So in the absence of oxygen, cells can regenerate NAD+ by using pyruvate itself as an electron acceptor. We're going to look at two specific examples of that. Lactate acid fermentation and alcoholic fermentation. And then in the presence of oxygens, then we can undergo aerobic oxidation and cellular respiration in the mitochondria, right, which is what we're going to look at as the main source of energy. So that's the pathway that gives us the gigantic stores of ATP. So when we look through fermentation pathways, keep those numbers in mind and compare those when we get to the end of cellular respiration for the difference in what we're producing in terms of energy. All right, so we can regenerate that NAD+ either in an anaerobic way in fermentation using pyruvate as an electron acceptor. So NAD+ picks up an electron and a hydrogen in that second phase of glycolysis. In order to reset, it's got to be able to dump that electron onto something else. In fermentation, it dumps that electron onto pyruvate in various processes. In respiration, we're going to look at aerobic respiration specifically, but in this pathway, pyruvate is not the electron acceptor, something else is. We're going to start off just looking briefly at the most common fermentation pathways. Those pathways that don't require oxygen. Two specifically, the most common in cells. Oh, first, fermentation. We looked at this as well, the definition. Series of oxygen independence, oxidation reduction reactions that oxidize NADH back to Na to regenerate NAD+. So we have to dump those electrons onto another molecule. So if we look at the last set of reactions in the glycolysis pathway, C3P, we generate our NADH. We talked in detail about the enzymatic reactions that promote that. We take that all the way to pyruvate. And then in fermentation, pyruvate is going to take on that electron. So the first one is lactate fermentation. The enzyme lactate dehydrogenase. All right, so here is our pyruvate. We're picking up that hydrogen onto that pyruvate to regenerate that NAD+. So at the end of glycolysis, we have pyruvate and NADH. We can go through the lactate fermentation pathway to generate NAD+ and lactate. This is the sort of thing that happens in your muscles. And then perhaps everyone's most favorite fermentation pathway, the one that generates alcohol. So this is a two-step fermentation pathway. We start again with pyruvate. We have a pyruvate decarboxylase. So we're pulling off a carbon. In the first step, carbon dioxide. And then in the second step, there's an alcohol dehydrogenase that can pull off that hydrogen and electron to make a molecule of ethanol. You see this in microbial organisms like yeast. Very useful pathway. All right, so we start in this case with pyruvate and NADH, two-step process. Enzymes involved, pyruvate decarboxylase, strip off the carbon dioxide. Alcohol dehydrogenase takes that electron hydrogen from the NADH to produce NAD+, ethanol, and carbon dioxide. All right, so it's oxygen independent. The most common ways are the production of either ethanol and carbon dioxide or lactate. This is the way that your book represents it. So in the absence of oxygen, pyruvate can take a number of fermentation pathways. And remember, out of glycolysis, we are getting ATP. We're getting a net of 2 ATP for every molecule of glucose. We put 2 in, we get 4 out, for a total net of 2. So as long as we can keep that NAD+ going, we still get 2 ATP out of our glucose through glycolysis. Not a lot of ATP, but we are generating some. There are lots of other types of fermentation pathways. Here are a few. I want you to be familiar just with that lactate fermentation and the alcohol fermentation. One's a one-step process, one's a two-step process, where that NADH gets regenerated to NAD+. All right, so anaerobic fermentation can keep cells going. It can keep NAD plus regenerated. So glycolysis can continue. You get a little bit of ATP out of it each time. But it's not a huge gain. Where we're going to see our huge gain in ATP generated is through aerobic respiration. So the rest of that pathway I've been showing you. So we're going to oxidize pyruvate. And the first thing we need to do is get that into the mitochondria. So instead of these anaerobic fast fermentation pathways, the alternate route in the presence of oxygen is for pyruvate to go into the mitochondria, be converted to acetyl-CoA, and go into the citric acid cycle. So if we look at aerobic respiration, the next step then is pyruvate, coenzyme A. We produce a molecule of CO2, a molecule of NADH, and we end up with acetyl-CoA. The enzyme pyruvate dehydrogenase does that. All right, so then we're ready for respiration. We're ready, we're in the mitochondria, we have the pyruvate in the right form. Let's generate a bunch of ATP. All right. You don't need to answer this question yet. I'm going to show it to you now because you will have to answer it later. So think about why aerobic respiration, why is oxygen so important? What role is it playing in this whole process? All right, so keep an eye out for when oxygen comes in to this whole set of linked pathways and what its role is. We'll come back to this question. I'm giving you a heads up on this one. Think about this. Watch for the oxygen. All right? So that first step that we just talked about, oxidation of pyruvate to CO2. So we're taking pyruvate into the mitochondria, and we're going to oxidize it all the way to carbon dioxide. And in the process, we're going to generate some energy molecules, GTP or ATP, as well as some electron carriers. NADH, we've already seen some of. A related molecule, another electron carrier, FADH2. All right, so also keep track of how many of these we generate. All right. We've got glucose in the cell. We've taken it through glycolysis to pyruvate and generated some ATP and NADH along the way. Now we're going to take pyruvate into the mitochondria, where it undergoes oxidation to acetyl-CoA. That acetyl-CoA is going to go into the tricarboxylic acid pathway cycle, and it's going to be oxidized to CO2. Here's where we're at. 2 net ATP from glycolysis, 2 NADH, 2 pyruvate. For each molecule of pyruvate, we get an acetyl-CoA, CO2, and an NADH. But we have to get that pyruvate into the mitochondria. So the rest of the pathways we're going to talk about happen in the mitochondria, and you have to keep track of where. So remember, the mitochondria has 2 membranes, which means that there are multiple spaces things can happen in. We have the outer membrane, we have the inner membrane, we have the space between the 2 membranes, and we have the matrix inside the inner membrane, and those folds of membrane we call the cristae. So different parts of this pathway, these linked pathways, happen in different areas of the mitochondria. For pyruvate oxidation to acetyl-CoA, that happens in the matrix. So pyruvate has to get across 2 membranes for cellular respiration to happen. Right? There are a series of porins. So these channel proteins on the outer matrix, or the outer membrane, that pyruvate moves through to the space between the two membranes. And then there's a pyruvate-specific symporter that's going to move it across the inner membrane. So there have to be two transport proteins for anything that's going to get all the way into the matrix. And then we are going to get to all these little purple light bulb things. Right, those are the proteins on the inner membrane where the electron transport chain happens. And we're going to look at that in detail as the last part of cellular respiration. All right, so that happens on the cristae. So pyruvate oxidation and the TCA cycle happen in the matrix. The electron transport chain happens on that inner membrane. All right, so let's move our pyruvate into the mitochondrial matrix. Here is another look at what's happening in pyruvate oxidation. So we're stripping off a carbon, CO2, electron, to go from NAD plus to NADH, and coenzyme A is picking up that acetyl group that's left. All right, so notice the difference here between what happens in fermentation, where pyruvate is picking up the electron, versus what happens in cellular aerobic respiration, where it's continuing to give off electrons to additional electron carriers. So we haven't regenerated NAD plus yet. We're still getting to that part. If we look at this coenzyme A, all right, it's going to look a little bit familiar. Again, we have as the base our nucleotide, adenine, another pyrophosphate bridge like we saw in our NADH. Then we have this pantothenic acid with a sulfhydryl group on the very end. All right, this is going to look familiar. We talked about a similar enzymatic reaction already, once in glycolysis. All right, so this sulfhydryl group picks up this acetyl group from pyruvate, forming that thioester bond. And it's going to carry that acetyl group into the tricarboxylic acid cycle. All right. So what are the products of pyruvate oxidation? Check in and make sure everyone's keeping track. From one molecule of pyruvate, what's produced? One molecule of pyruvate.
Oh.
Is it not working?
Oh, bummer.
Darn it. My clicker base just does not like Monday mornings. All right. We'll stop that then. All right, so what are the products of pyruvate oxidation for one molecule of pyruvate? One of everything. One NADH, one acetyl-CoA, one CO2. If I had said, starting with one molecule of glucose, what's produced after pyruvate oxidation, what would it be? Two of everything. All right. So here's where we're at. Everyone got their totals all in sync? We're going to take now that acetyl-CoA, two of them, and we're going to run them through the TCA cycle. So if we look in detail at the citric acid cycle, let me just say up front, I'm not going to expect you to know these structures. What you need to keep track of, right? There are 8 steps in this cycle. So where you get out your energy, right? And your electron carriers, what's happening at each step. So in this schematic, right, these gray balls represent carbons in the molecule. So pyruvate is a 3-carbon molecule. We strip off a carbon dioxide so that we have a 2-carbon acetyl-CoA. That 2-carbon acetyl gets joined with a 4-carbon molecule, form a 6-carbon molecule. Then we go through a series of molecular rearrangements and enzymatic reactions where we strip off carbon dioxide, we strip off a carbon dioxide, so that we're back to a 4-carbon molecule. And in the end, it's a cycle because we start off, or we end up where we started off. Right, with that same oxyl, I can't ever say it, the oxaloacetate, it's hard to say. We start off with that same thing that we combined with our acetyl-CoA to start the process. All right, so if we look at this, there are 8 chemical reactions in this cycle that go around and end up at the same place that we started. It's a series of carboxylic acids that are oxidized. All of this happens in the mitochondrial matrix. So it's called the TCA cycle, not because it's the citric acid cycle, but because it's the tricarboxylic acid cycle. All right, if we look at where we start with citrate, 3 carboxylic acid functional groups on that. Also known as the Krebs cycle, after the person who first described it. So it's referred to in multiple ways. And then we're starting with this citrate. How do we get to citrate? Acetyl-CoA plus oxaloacetate. Our 2-carbon plus our 4-carbon gives us our 6-carbon citrate to start off the cycle. So that very first reaction, we dump our acetyl into the cycle by combining it with oxaloacetate to citrate. Now notice in citrate, the blue and the red carbons. The red carbons are coming from that acetyl group that's coming from pyruvate. The blue carbons are the ones that are going to get converted to carbon dioxide throughout the cycle. And through the molecular rearrangements in the cycle, we're moving those carbons from the acetyl group into those positions that will get turned into CO2 the next time around. All right, so step 1, oxaloacetate, acetyl-CoA to produce citrate. Step 2, the molecular rearrangement. Again, right, we want to try to get things into favorable bonds for the reactions that we're trying to produce NADH and ATP. All right, so we've got a switch in the position of the hydroxyl from citrate to isocitrate. And then in step 3, we have our first electron carrier. We strip off a CO2, one of those blue CO2s. Oops. Go back. All right, so we produce NADH and CO2. Then we go from isocitrate to alpha-ketoglutarate. All right, so it's in step 3 where we get our first carbon dioxide and our first NADH. Step 4, we strip off another carbon. We go from alpha-ketoglutarate to succinyl-CoA. And we get a CO2 and another NADH. And we have that coenzyme A coming back in. So notice we've now gotten rid of both of those blue carbons as carbon dioxide, and we're left with those red carbons that came from pyruvate through acetyl-CoA. All right, in step 5, we generate a molecule of a nucleotide triphosphate. It can be either GTP or ATP. Both serve as energy for the cell. Bless you. We're going from succinyl-CoA to succinate. We have looked at a coenzyme reaction before in glycolysis. This one functions similarly. So let's just take a look at that process briefly. Should start to look familiar. So we're going from succinyl-CoA and phosphate and GDP, where we're starting and we're ending with coenzyme A, our succinate, and GTP. How do we get that phosphate onto the GTP? All right, everyone remember back to that process? No, let's take a look at it again. It happens the same way. Phosphate attacks that coenzyme A bond, where that thioester is. It knocks off that coenzyme A. So we're putting the phosphate onto our substrate first. What kind of bond have we just created? You don't have to answer it since clicker is not with your clicker. Just answer out loud. Draw it out. Right? So if you draw out your group, an anhydride, right? And remember, we talked about the free energy when you break that anhydride bond. When we talked about glycolysis and the molecular conversions we were doing there. So we put our phosphate into an anhydride bond on our substrate. And then from there, the enzyme can break that bond and put that phosphate onto GDP to make GTP. So there's that covalent intermediate. Create the bond with enough energy to get that phosphate onto our GTP or ATP. So what kind of phosphorylation is that then? Shaking your head, you're not sure? What do we do with our phosphate first? We attach it to the substrate. So it's substrate phosphorylation. What does what mean? So substrate is the thing that is undergoing the enzymatic reaction in the cycle. So at this point, our substrate is succinyl-CoA. We're adding a phosphate to that to knock the CoA off, and then we take the phosphate to ATP or GTP. So that phosphate is coming from the substrate. So we have to put it on there first to get it into that anhydride bond, and then we can put it onto GTP or ATP. All right, so even though it's not direct, it does form that intermediate step with our substrate, therefore it's substrate level phosphorylation. We haven't looked at what oxidative phosphorylation is yet. We'll look at that in the electron transport chain for comparison. All right, so just to show it all in one slide, so we have our succinyl-CoA synthetase, that enzyme. Phosphate attacks that thioester bond. We get CoA, and we have our succinyl now with a phosphate on it. That gets transferred to our enzyme to produce succinate, and then that enzyme can transfer that phosphate to a nucleotide. All right, so we're undergoing these rearrangements in order to get them in bonds where we can put them onto the right molecule. All right, so we're going from succinyl-CoA to succinate. Step 5, we produce either GTP or ATP by substrate level phosphorylation. So we put that phosphate onto the substrate first. All right, step 6, we're almost there. There are only 8 steps in the cycle. We go from succinate to fumarate. We produce FADH2. So this is flagon adenine dinucleotide. Right? Similar to that nicotinamide adenine dinucleotide. It can pick up electrons, so it's an electron carrier. Why FAD versus NAD plus? It can carry 2. And notice here we're stripping off those hydrogens from a carbon-oxygen. Here we're stripping them off of a carbon-carbon. All right, so there's a difference between these molecules and how they can be used relative to the energy and the bonds that that electron is coming from. All right, so produce our FADH2. First time we've seen that. Step 7, fumarate to malate. All right, we add in a molecule of water for a structural rearrangement. We break that double bond. Step 8, malate to oxaloacetate. Back to where we started, and we produce one last NADH. All right, notice now that those red carbons, that top carbon now is a blue carbon, and is going to be stripped off as carbon dioxide in the next round. We've gone back to where we started, completing the cycle. Bless you. And we're ready to pick up another molecule off of that acetyl-CoA. All right, so that's the cycle. You don't need to know the structures. I want you to know where things are produced. I want you to know it's substrate level phosphorylation. I want you to know how many things are produced. So here's a look at that simplified view again. Know that it's a cycle. Starts off by combining a 2-carbon molecule with a 4-carbon molecule. Get to that 6-carbon molecule. We strip off 2 carbon dioxide first thing. Then undergo a series of molecular rearrangements to dump electrons onto our electron carriers. All right, have you counted up? So 4 molecule of pyruvate that goes into the TCA cycle. What is produced? How many CO2?
3.
We're going from a 6-carbon to a 4-carbon.
I think we took the one we mentioned before.
Ah, very good. I even forgot to count that one. All right, so if you're coming from pyruvate all the way through, then it's 3 CO2. If you're going just from acetyl-CoA through the TCA, then it's 2 CO2. How many NADH? 3 out of just the TCA cycle. How many FADH2? 1. How many GTP? 1. All right. What about if we were starting with the product of glycolysis? What would be the product of just TCA? Double that. What about if we started all the way at glucose? All the way back to the beginning. What have we produced up until this point?
We just have to add on 2 ATP and then 2 NAD. Like starting from the glucose from the total number you put add on.
Remember, we're talking about everything that happens. Hand it up. We'll give you 1 second. Glycolysis, pyruvate oxidation, TCA cycle. Everything we've produced so far.
6 CO2, 10 NADH, 2 FADH2, 4 GCP.
Six CO2, 10 NADH, 2 FADH, 4 GTP or ATP. All right, we can break that down. Maybe make it a little bit clearer where they're coming from. So 2 ATP out of glycolysis, 2 NADH, 2 pyruvate. We oxidize that pyruvate to produce, remember there's 2 of them, 2 NADH, 2 acetyl-CoA, 2 CO2. And we put that acetyl-CoA into the citric acid cycle and get out 6 NADH, 2 FADH, 2 ATP or GTP, and 4 CO2. All right? Everyone see that? They're still counting up. We start all the way at glucose, count everything that happens in glycolysis, and then everything that happens after that twice. 1 for each molecule of pyruvate. What we haven't done yet, make much ATP. All right, so we're almost there. We're going to get that generation of ATP finally in the last set of reactions in this whole series we've been going through in the electron transport chain. All right, so if you look at what we've generated so far, we've generated a little bit of ATP. Mostly what we've generated, though, are those electron carriers. Tells you we're going to do something with those electrons. It's going to generate our ATP for us. All right, so here's where we're at. Out in the cytosol, in the matrix of the mitochondria. The last thing we're going to talk about is that electron transport chain. So now we're going to feed all of those electron carriers that we've produced into the electron transport chain. Notice that this is where we're going to use our oxygens. We're going from oxygen to water. So what does that mean about what oxygen is doing? And we're going to generate a bunch of ATP. All of this happens on the cristae membrane. So we're on a membrane. So we're going to be moving those electrons back and forth across the membrane to create electrochemical gradients that are going to drive this process for us. So the electron transport chain, here's where we get to oxidative phosphorylation. We're involving oxygen finally in this whole process. This step converts those energy carriers to ATP. And it requires oxygen. So the electron transport chain, we're going to look at in detail first thing on Wednesday. Membrane-embedded proteins that are going to accept high energy electrons. And by this series of passing them from protein to protein, they're going to sequentially lower the energy state of that electron. So finally it gets dumped onto oxygen. All right, and then that energy is used to generate a gradient that's going to drive the generation of ATP. We'll start there on Wednesday. Okay.